All right, everyone, welcome back. Next up, we have Tonix Pharmaceuticals. Tonix Pharmaceuticals trades on the NASDAQ under the symbol TNXP. Tonix is a clinical stage biopharmaceutical company focused on discovering, licensing, acquiring, and developing small molecules and biologics to treat and prevent human disease and alleviate suffering. Tonix Pharmaceuticals CEO and President Seth Lederman is here with us today. Welcome, Seth. There, unmute yourself.
There.
There
Welcome.
you
Can you
go.
hear me now?
Yes, perfectly.
Okay.
All right, whenever you're ready, Seth, and maybe turn it up just a little bit or come a little closer to the mic so we can hear you better.
Okay.
Thank you,
Well,
sir.
thank you everyone for joining this presentation. I'm Seth Lederman. I'm CEO of Tonix Pharmaceuticals, and we have a lot of exciting news. This morning we had a big press release about a vaccine program, and we have a lot more news coming up. So this is a very exciting time for you to look at Tonix. We trade on NASDAQ, TNXP. TNXP is our ticker. So we are a public company. Get control of this. We're a public company, so this is our disclaimer on forward-looking statements, and I refer you to our 10K, which we just filed on Monday evening, so it's very current. So the 10K and other documents filed in the SEC are a more complete information about the company. Tonix is a clinical stage biopharmaceutical company, and we have two major areas that we work in. One is central nervous system conditions, and the other is immunology. So first I'll show you our central nervous system pipeline. And shown here is our central nervous system pipeline. And highlighted in yellow is our fibromyalgia program, which is our lead program. That is in phase 3 development. And we just announced a positive phase 3 study in December of last year. So this is now in mid-phase 3. There's one more phase 3 to go. And we recently, on Monday of this week, announced that that trial is 50% enrolled. So we expect to have an interim analysis from the second confirmatory phase 3 trial in the third quarter of this year. And as I just mentioned, the cohort that will be analyzed in the interim analysis has been enrolled. I'll tell you much more about this program. It's our lead program. Now that drug, TNX-102SL, is in development for 4 different indications. Fibromyalgia is the lead, but it's also in development for post-traumatic stress disorder, agitation in Alzheimer's disease, and alcohol use disorder. And we have active INDs for all 4 of those indications. Now we have a robust pipeline in CNS. I will briefly mention some of these, and we'll have just a slide or two on the other programs. I'm going to spend most of the time talking about the lead program in fibromyalgia. So in addition to the 102SL programs, we have TNX 1300, which is an antidote for cocaine intoxication. It's a biologic. It has FDA breakthrough therapy designation. It's already been in one phase 2 study, and it's going back into another phase 2 study imminently. Below that, TNX 1900. That is a program to treat migraine. It's an inhaler, an intranasal applicator device. The drug is oxytocin, and that is going into phase 2 this year. Below that, we recently announced the in-licensing of the technology underneath TNX 2900, which is a potential treatment for the orphan disease called Prader-Willi syndrome. So we just made that announcement about a month ago, and we will update. I'll have a slide for you later about Prader-Willi. TNX 601CR. This is a potential treatment for depression, and TNX 1600 is a earlier stage treatment for depression, PTSD, or ADHD. So wait, I skipped one slide. There we go. So here's our immunology portfolio. And the lead immunology program is highlighted in yellow, and that is TNX 1800. That is a potential COVID-19 vaccine. We just announced update this morning, which explains the activity in our stock. Very exciting news. We vaccinated non-human primates and announced last year that they had a good immunological response, a strong immunological response to COVID-19. And just this morning, we announced that after challenge with live CoV-2, that's the virus that causes COVID, the animals, the vaccinated animals had undetectable CoV-2 virus in their mouth, their oropharynx, and in their lungs, their lower airways. So a big announcement this morning. I'll talk more about that program. Let me quickly run down some of the other things we have. We have TNX 2100, which is a skin test for COVID-19 T cell reactivity. I'll have a slide on that later. And then let me just focus on TNX 1500, which is a monoclonal antibody to prevent organ transplant rejection. So I'll have a slide on that. So now let me turn, I'm going to spend most of this presentation talking about our lead program. The lead phase 3 program is TNX 102SL for fibromyalgia. So here's some background on fibromyalgia. It's a chronic condition. It is a pain disorder. It is a chronic pain disorder. So people with fibromyalgia experience pain all over their bodies. And it's relatively common. About 10 million American adults suffer from fibromyalgia, and about 90% are treated with some form of drug. But it's actually an interesting market for a new drug to enter, because even though there are 3 FDA-approved drugs to treat fibromyalgia, patients remain dissatisfied. And you can see that in a number of ways. One is that people on drug treatment switch and rotate between the approved therapies and also unapproved therapies. Another attribute of the fibromyalgia market is that people are taking more than one medicine. That's called polypharmacy. So you can see here that on average, an American adult fibromyalgia patient is taking almost 3 medicines at the same time to treat fibromyalgia. And then finally at the bottom, dissatisfaction. Only about 43% of fibromyalgia patients are satisfied, which means that more than half are not satisfied. So here we give an idea about what we think would be the ideal profile for a new medicine, something that would treat fibromyalgia as a syndrome, and I'll go into that more later, something that's well tolerated with low discontinuation rates, because fibromyalgia is a chronic disease, as noted in the bottom bullet. So something that's well tolerated, works over a long period of time, and has broad activity. These would be 3 attributes of a successful product. Another very important element of this is the risk of addiction. About 1/3 of fibromyalgia patients in desperation result to using chronic opiates. So a new product that had no addictive potential would be very valuable. So here's a description of our product, TNX 102SL. And let me explain why we think that this fits many attributes of an important new product. First, let me tell you what it is. It is a reformulation, a sublingual tablet. That means a tablet that goes under the tongue. It's a sublingual tablet formulation of cyclobenzaprine. Cyclobenzaprine has been marketed in the United States for many years, but it's been marketed in a form where the pill is swallowed. So a sublingual tablet is a different route of administration. And there are several potential advantages of a sublingual formulation, and particularly ours, because it's designed so that the cyclobenzaprine is absorbed through the skin under the tongue. And that's called transmucosal absorption. And that means that the cyclobenzaprine doesn't have to go into the stomach to enter the bloodstream. The advantage of that is by being absorbed in the mouth, it avoids getting metabolized by the liver. So this is a big advantage, that our sublingual tablet of cyclobenzaprine can be absorbed in the mouth, transmucosal absorption, bypasses the liver, and goes to the brain where it does its work. Now, it's also important that cyclobenzaprine has no recognized abuse or dependency concerns. Again, something I mentioned is very important for a drug to treat a chronic pain syndrome. Here's a schematic diagram of the relief study. This is the study that we completed last year and announced the positive results of in December of 2020. As you can see, there are two arms to the study, drug in blue and placebo in red. We targeted about 470 patients and we enrolled 503. The primary endpoint was daily pain severity, which is what FDA asks for the primary endpoint of fibromyalgia studies. And as I mentioned, this is a positive study, so it's the first of two studies that are required to get NDA approval. Now, another reason to look at this schematic is that the new study that we're enrolling, called the Rally study, is essentially identical to the design of this study. Since we had a positive study, we don't want to change anything, except the one difference between the Rally study, the new phase 3 study, and this is that we are enrolling more patients in the Rally study. This relief study targeted 470 patients. The Rally study is targeting 670. Here's the top line results from the relief study. And if you see to the right, the p-value, that's the most important number that the FDA looks at, the p-value was 0.01. That's a strong p-value. The threshold for FDA is typically less than 0.05. So 0.01 was very encouraging to us. And this was the pre-specified primary endpoint of the relief study. This is another way to analyze the same data, and this is called a pain relief responder analysis. So it's the same data that led to the 0.01 on the last slide, but it's another way that people have analyzed fibromyalgia pain data in other studies. And we're just showing it here to show that by this other method, the p-value from our last phase 3 study was 0.006. So again, quite a positive study. Now, in addition to the primary analysis of pain, we looked at a number of other things, and that's because fibromyalgia is more than just a chronic pain disorder. It is a syndrome that has many different elements. The three most important elements to patients from our market research and from the literature are pain, sleep disturbance, and fatigue. And if you look at the bottom, of this slide, the promise fatigue had a p-value of 0.018, and two measures of sleep quality. The daily sleep quality diary was 0.001, and the promise sleep disturbance score was 0.001. So we had strong effect sizes on pain, sleep, and fatigue. Those are really the important measures that matter to fibromyalgia patients. Now, we were also had strong activity in the fibromyalgia symptom domain by the FIQ, the Fibromyalgia Impact Questionnaire, and also the function domain. In the patient global impression of change, we had a near miss. If you see, the p-value was 0.058. So that was the one element of the study that was not positive. We don't really understand that. In prior work, we've shown very strong PGIC results. But nonetheless, as I mentioned, we've shown with TNX 102SL in this study, it had broad spectrum activity, pain, sleep, and fatigue, and it hit the primary efficacy endpoint. It was also well tolerated. And this table shows the side effects, and typically tables like this, and this one does, shows side effects that were present in more than 5% of any group in the study. So you can see the only systemic adverse event that showed up on this chart was somnolence or sedation. And that means that the patients were feeling sleepy during the day. But this is at a relatively low incidence. You see it was 5.6% incidence in the drug group. And this is counted as positive if they experience this symptom anytime during the 14-week study. At the bottom part of this table, it talks about local administration site reactions. As I mentioned, this is a sublingual tablet. It is designed to break apart very quickly in the mouth and to release the drug into the skin, into the tissue underneath the tongue. So these are basically sensations that the patients have during this process. The most frequent is described as tongue numbness, but when this was reported, they were transient, self-limited events, and they did not happen consistently in the patients who experienced them for the most part, and they were only experienced by a minority of people taking the medicine. Fibromyalgia, as I mentioned, has 3 approved drugs, and they were developed by Pfizer, Lilly, and AbbVie, actually by Forest Labs. It was bought by AbbVie. But Pfizer marketed Lyrica, which is now generic. Lilly marketed Cymbalta, which is now generic, and AbbVie markets Savella. Lyrica from Pfizer was a very successful drug for fibromyalgia. As a matter of fact, it had sales of about $5 billion per year when it went off patent. And when it went off patent, it was Pfizer's largest drug product. Lilly marketed Cymbalta and also had a peak sales of around $5 billion. And when it went off patent, it was Lilly's largest product by sales. So these examples show that new medicines in fibromyalgia can be well received by patients. Now those drugs are generic now, and so the big question is, how will our medicine stack up to generic Lyrica or generic Cymbalta? And I think that we win in 2 potential ways. One is we have broad spectrum activity. And I mentioned we have effects in pain, sleep, and fatigue. At best, Lyrica had effects in pain and sleep. And at best, I think that the Cymbalta really had effects in pain. So we believe that we match up well because our medicine covers the 3 areas that matter most to patients. Another way that I think that our product has the potential to get market acceptance is tolerability. Lyrica is hard for many patients to take, and there's literature and patient experience that many patients who take Lyrica can't tolerate the dose that showed activity in the studies. So with that kind of a restriction, it seems that patients who can't tolerate the full dose may not be getting the benefit that was described in the study. And Cymbalta has other issues relating to tolerability, but again, we think that our tolerability profile has the potential to differentiate us in these other products in the market. So here are what we think are the next steps for this program. As I mentioned, we are enrolling in the second phase 3 study that we call Rally. It has the same protocol design as Relief. And Relief, as I mentioned, had positive results at the end of 2020. But the one difference between Rally and Relief is that we have approximately 200 more patients targeted in Rally. The enrollment began in September. We just announced the full enrollment of the first half of the patients. We expect to have interim results in the third quarter and have top-line results before the end of the year. So this is obviously our lead program, phase 3, mid-phase 3, with top-line results in the second confirmatory study expected this year. Now, following positive results from Rally, we believe we can file a new drug application in 2022. We've already completed the long-term safety exposure studies, and we've already completed the development of GMP manufacturing processes. So this is the intellectual property portfolio for 102SL. As you can see, several patents have issued in the top. This shows the global patent portfolio, but if you just look at the top bullet, issues several patents have issued in the United States, and we have protection until 2034 or 2035. So with that, I'm going to turn to the COVID vaccine story. And I'll remind you that we just reported positive results from vaccinated monkeys this morning. But now let me set up the story about that. There are 3 emergency use authorized vaccines available in the United States, but there's a big question about durability. How long are vaccinated people protected? And also, how long are people protected after they recover from natural COVID? COVID is closely related to some viruses that cause the common cold, that are called common cold coronaviruses. And the immunity to those viruses is notoriously short-lived. So while we're hopeful that we're going to get herd immunity through people recovering from COVID and from vaccination, it's a big question about how long that immunity will last them. Another question is protection against forward transmission. That means, will someone who's vaccinated or someone who's recovered still be able to spread it to other people? Another issue is biomarker protection. We're vaccinating a lot of people, but we don't know, for the most part, who's protected. The variants are a big problem. I'm sure you've heard about the British variant, the South African variant, the Brazilian variant, the Californian variant. So all of these have shown greater infectivity, the potential to make people even sicker than the original Wuhan CoV-2. And also, it questions the ability of the existing vaccines to protect against them. So with that, let me tell you how we are addressing these. We're developing TNX 1800. It's a novel COVID-19 vaccine. It utilizes our proprietary horsepox vector as horsepox virus as the vector. Now, we've engineered horsepox to express the spike protein from SARS-CoV-2. And we, as I mentioned at the outset, we announced some results of animal testing, the immune response results last year in the fourth quarter. And just this morning, we announced that non-human primate CoV-2 challenge testing had positive data, and it appeared to decrease, for the most part, to undetectable, the amount of virus in the mouth and lungs of the vaccinated animals. So we've previously announced a partnership with Fujifilm Diosynth to manufacture the vaccine, and getting a manufactured vaccine is the key gating item right now, we believe, to starting a human study, which we expect to do this year. Now, just one slide on TNX 2100. Our work on the vaccine on TNX 1800 has convinced us of the importance of T cell immunity to COVID-2 to stay healthy. And not all vaccines induce T cell immunity equally. Our TNX 1800 vaccine is designed to elicit a predominantly T cell response. But T cells are also useful in establishing whether someone has been infected. So you may know that there are two basic types of the immune response: antibodies and T cells. T cells are known to last many years, even decades, whereas antibodies fade over a period of 6 to 9 months and are usually low or gone a year later. So this raises important questions about how to understand herd immunity in the United States and also understand who has been infected. Because now that we're past the 1-year mark where many people started getting infected, many of them will no longer have antibodies. So the test that we've developed is to use peptides from SARS-CoV-2 and to inject them into the forearm, this aspect of the forearm, just like the test for tuberculosis called PPD, or a Mantoux test. So in this test scenario, someone comes to the doctor's office and gets a small injection in the skin of peptides, and then 24, 48, 72 hours, typically 48 hours later, they would come back to the physician office and have the physician look at the area that's called indurated, which means hardened and raised. And that's an indicator of T cell immunity. And it's an indicator of T cell immunity for tuberculosis. And with the peptides that we've designed, we expect it will be an indicator of immunity for COVID-2. Now moving to our CNS portfolio, our central nervous system portfolio, we have TNX 1900. This is an intranasal oxytocin formulation. Intranasal means that it's a nasal spray, it's injected up into the nose, and it gets absorbed in the back of the nose and brings the oxytocin into the brain. So we believe that intranasal oxytocin has potential in treating migraine. The company from which we acquired this had done a foreign study and had also done a US study under an investigator IND, and both of them had encouraging data. But we have now taken it over. We're developing a stable pharmaceutical grade formulation, and we expect to take this back into the clinic for a phase 2 trial in the 3rd quarter of this year. And for those of you who haven't followed the migraine market, it's a very exciting time in the migraine market because there's several new biologics. For example, Teva has Ajovy, and there's some other similar products. So there's a lot going on in the treatment of migraine, and we think that this could be a very important component and a completely different mechanism of action from the anti-CGRP antibodies. So because we were working on intranasal oxytocin, we also saw the opportunity to develop it as a treatment for the orphan or rare disease condition called Prader-Willi syndrome. So we were fortunate to be able to license technology that supports the treatment of Prader-Willi syndrome with our intranasal oxytocin. And we acquired this technology from essentially what would be the equivalent of the French NIH. It's called INSERM, INSERM. So we recently announced that, and we look forward to updating the market soon once we've nailed down our plans. But basically, what the scientists at INSERM found is that by treating mouse models of Prader-Willi with intranasal oxytocin, or in their case, injected oxytocin, you can really make substantial improvements in the disease, and not only treat aspects of the disease, but also if you start early enough, you can reverse some of the developmental problems that happen along the way. Another drug from our CNS portfolio is TNX 1300, or cocaine esterase. This is a recombinant protein that encodes an enzyme from a bacteria that rapidly disintegrates cocaine. So in a phase 2 study, our drug, this biologic, was injected into cocaine abusing volunteers, and then they were injected with cocaine, and this enzyme rapidly degraded the cocaine. So we are now imminently going to start a phase 2 open label study of TNX 1300 in the emergency room setting to see whether this can function as an antidote in emergency rooms, similar to the effects that it had in the controlled environment of a laboratory. And then the final program that I'm going to highlight from our diverse portfolio is TNX 1500. This is a monoclonal antibody, a biologic that is in development to treat transplant allograft rejection. Allograft is when one human donates an organ to another human. It's the most common type of transplantation. So there have been 2 prior generations of CD40 ligand before, both of which made it to the clinic. In each case, people were very impressed by the activity of the drug, but were concerned about a side effect. And what we've done is we've redesigned a monoclonal antibody CD40 ligand. And we believe that we've maintained the efficacy by modifying the constant region that on the slide on the right is called the Fc gamma R region of the Fc region, that we can deliver this efficacy with reduced issues on tolerability. So we just announced our K earlier this week. And so as of December 31st, we had $77 million. We've done 2 offerings in 2021 already that have raised gross about $110 million. And here's our share count, 324 million at the end of Monday. I have a sluggish slide advance. Let me see. There we go. Okay, so here are milestones. As I said at the beginning, we have an action-packed year. As you see, the new checkbox, the third one down, is checking the box for non-human primate positive efficacy data that we just announced this morning. I'm not going to go through all of these. This deck, by the way, is posted on our website. But let me just call out some key points for you to look out for this year. A key thing in the third quarter will be the interim analysis on the confirmatory phase 3 study of TNX 102SL for fibromyalgia. So to repeat that, in the third quarter, interim analysis for 102SL for fibromyalgia. In the fourth quarter, top-line data from 102SL in the confirmatory phase 3 study. So those are really the highlights. We have a number of programs, as I've told you. I've just taken you through a number of interesting programs. So we expect to have news throughout the year. But at this point, I think I have about 13 minutes left. So I'm going to turn it back to the organizers and hopefully take some questions.
Great job, Seth. We do have quite a few questions.
Great.
All right. So can you discuss the mechanism of action for TNX 102SL and how it's differentiated from the approved products for fibromyalgia?
Thank you. That's an excellent question. The TNX 102SL, the easiest way to understand it is that it improves sleep quality. And as I mentioned, fibromyalgia patients have a sleep disturbance, and many of them report that if they can get a good night's sleep occasionally, that they feel better. So the idea behind this, which is now validated by a positive phase 3 study, is that a medicine taken every night at bedtime can improve sleep quality, and improving sleep quality can lead to an improvement in pain, so a reduction in pain. Now, the drug has a completely different mechanism of action from the 3 approved fibromyalgia products. Our drug, cyclobenzaprine, interacts with 3 neurotransmitter receptors in the brain, and each one of these is involved in propagating and perpetuating the sleep disturbance. And so when cyclobenzaprine is in the brain, it blocks these 3 receptors, and it allows the affected person to get improved sleep. So it is a new mechanism of action in fibromyalgia. As I mentioned, it is non-addictive, and it partially has those 2 properties because it attacks fibromyalgia indirectly. It attacks it by improving the sleep problem, after which a natural healing process is believed to start.
Thank you for that very thorough and knowledgeable answer, Seth. Next question is, assuming that the RALI study is positive for fibromyalgia, when do you expect you might receive FDA approval and be able to launch?
Thank you. If the RALI study is positive in the fourth quarter, we anticipate being able to file the new drug application in the first half of 2022. So to make that happen, we have already begun the process of preparing the NDA. We feel that since the RALI study is basically a repeat of the successful relief study, that it's worthwhile to go at risk and begin preparing the NDA. So if the NDA is prepared, is submitted in the second quarter, in the first half of 2022, we expect that it would get FDA approval in the first half of 2023.
Perfect, thank you. What is your commercialization strategy for fibromyalgia?
Like many emerging biotechs, I think our preferred strategy would be to find an excellent commercialization partner, particularly a big pharma. As I mentioned, the Lyrica and Cymbalta were very successful commercial products. And for those of you who watch TV, you may remember that Lyrica and Cymbalta were heavily promoted on television for fibromyalgia. They had other indications, but fibromyalgia seemed to be the indication for which Lilly and Pfizer decided to market these products. So we do think that this has the potential to be commercialized by a big pharma. As I mentioned, about 10 million United States adults. But it's hard to predict where they will be, whether it will fit into their portfolio, et cetera. So although our plan A would be a commercial partnership, Plan B is that we could market it on our own, at least for a limited time. And sometimes that works to establish the market potential of a product. So, for example, some of you investors may remember Medivation, which initially didn't get a product. They marketed their product on their own for a while, and then were rewarded by a significant takeout from Pfizer. So I think Plan A would be to get a commercial partnership, and Plan B would be to market it on our own.
Okay, perfect. There are 3 COVID-19 vaccines in the EUA and the US. How is yours different?
Great question. So the 3 EUA vaccines, first of all, I've gotten my first dose of the Pfizer vaccine. I think that the mRNA vaccines, were developed at remarkable speed. And I'm so grateful to the scientists that worked on them and all the people at Operation Warp Speed in the Army and other people that got these vaccines developed and now that they're being distributed. So I'm a big believer in vaccines, and I encourage everyone who's eligible, when their time comes up, to get one of the 3 EUA vaccines based on what I know now. But I think that there are limitations. And as I mentioned in our presentation, the biggest question is durability. These mRNA vaccines are from a completely new technology. We really do not know if they will only provide protection for 6 months, for 9 months. We don't know what it will look like a year later. The Johnson & Johnson Ad26 vaccine is a somewhat tested technology. It was half of their Ebola vaccine, but again, it remains a fundamentally novel technology. And again, we just don't know the durability of protection. So we have designed our vaccine to elicit T cell immunity. T cell immunity is longer lasting than antibody immunity. And also, other people have shown, and we have speculated, but our analysis is that T cell immunity will have broader protection against the new emerging variants of CoV-2 that are coming up, because the epitopes for T cells, the epitope means the part of the protein that the T cell sees, seem to be mutating less than the epitopes that antibodies see. So I think those are the 2 considerations. One is, what is the durability of the existing warp speed vaccines? And the second is, how will these vaccines be affected as the virus mutates? Now, our technology, while the warp speed vaccines, the EUA approved ones, are of completely novel technology, ours is based on the oldest technology in the vaccine world. And I can say that pretty clearly because the very first vaccine was called Vaccinia in 1798, and it was developed by Edward Jenner, a physician in England. And we believe that our vector, the horsepox virus vector, we believe that that is closely related to the vaccine that Edward Jenner used. Now, Edward Jenner's vaccination program was very successful, and it led to the eradication of smallpox. Smallpox is the only disease ever eradicated. So we believe that the attributes of Edward Jenner's vaccine, the durable T cell immunity, can be translated into a COVID response. And that combination could be very important. Now, another way to think about the different vaccines is, they're different vaccines for different periods. I think that the mRNA vaccines and the Ad26, again, remarkable achievements, but they were first generation vaccines. COVID-19 is going to be with humans, I predict, as long as humans are on Earth. It will be endemic. We already know that it has animal reservoirs. It's extremely contagious. So I think that, like other conditions, like measles, mumps, and rubella, humans are going to have to exist or coexist with COVID-19. So ultimately, I think it will become a childhood vaccine, the same way that children in the United States are vaccinated with the MMR, the measles, mumps, rubella vaccine. So I believe that after the pandemic, this will be a childhood vaccination. And I believe that our candidate would be a very strong candidate to induce multi-year, maybe several decade protection against COVID-2. So thank you very much for that question.
All right. Let's see. If we have a few more questions, and we will send all of these to you so you can answer. A quick question: what is your current cash position?
Well, as I mentioned, we just put out the 10K on Monday, and we had $77 million in the bank at the end of 2020. In 2021, we've raised gross $110 million in two different financings. One was at 80 cents, one was at $1.20. So, given where we finished 2020 and our raises we've done this year, I think that we're in a solid financial position.
Wonderful. Well, we do have quite a few questions that might take a little bit longer, so we're going to send them directly to you, Seth, so you can answer at your convenience. But this was a fantastic, very knowledgeable presentation, and thank you so much for all of your hard work, and we look forward to following the updates of your company.
Thank you very much, and thanks to everyone who participated and for the questions.